Hopefully that's recording. So we talked about G protein coupled receptors. So what do you think is a big takeaway from G protein coupled receptors? Either something you already knew, but maybe we fine-tuned it, or something that was new to you. Did anyone learn anything new on G protein coupled receptors? Yes, Tyler. Okay, very good. The receptor is not an enzyme. What part of the whole system is an enzyme? Okay, so the G protein, the alpha subunit of the G protein is enzymatic. What chemistry does it do? So it's dephosphorylation. Okay, so the alpha subunit of the G protein, remember, does dephosphorylation of that GTP into GDP. Okay, so that's enzymatic. So it's just a GTPase. What other part of the whole system is an enzyme? Well, right, so the G protein activates another enzyme. What enzyme is that? Adenylyl cyclase. So that's the enzyme that turns ATP into cyclic AMP. Okay, so there are two enzyme components to the G protein coupled receptor, but the receptor itself is not an enzyme. Okay, and so what maybe is a takeaway from the acetylcholine receptor? Either something you already knew that probably is the most important thing to take away, or something new that you learned. Okay, how about that acetylcholine travels a very small distance, but it enables signal transduction over a very long distance. Okay, so acetylcholine only goes from the exit point of one cell to the receptor at the adjoining cell, but that small distance that molecule travels to its receptor enables signal transduction through the whole length of that next neuron, and then it continues the signal. Okay, so acetylcholine only travels a small distance, but enables long-range signal transduction. Okay, and so the next class of enzymes that we'll talk about are what are called receptor enzymes. And so the insulin receptor is one example of this. So a receptor enzyme is itself an enzyme. So it both binds the ligand, so the insulin receptor will bind insulin on the outside of the cell. Okay, so here's insulin binding on the outside of the cell. And what insulin binding triggers is activation of the enzymatic activity on the inside of the cell. And so what happens on the inside of that insulin receptor, the cellular part of the insulin receptor, is there's a domain that is a tyrosine kinase, so phosphorylation on tyrosine residues, right? So what does that phosphorylated tyrosine look like? So if this is a tyrosine, with an H there, right? So now we're going to have a phosphate group attached there. So now we have a phosphorylated tyrosine, and that is going to change the properties of the enzyme. Okay, so the receptor itself becomes autocatalytic, so it turns on that kinase activity, and there are three tyrosines that now get phosphorylated on the receptor itself. But then what that does is it also triggers phosphorylation of other things inside the cell. Okay, so it's this cascade of activation by phosphorylation. And the enzyme itself gets phosphorylated. But what that does is it opens up, so here is the tyrosine kinase domain, so phosphorylation of those three tyrosines on the inside of the receptor itself, conformational change, going to open an active site for another protein to come in and get phosphorylated. So phosphorylation of the receptor makes it able to phosphorylate other proteins. So now that triggers this phosphorylation cascade. So again, here's the insulin receptor, dimeric on the inside of the cell. So each monomer has three sites that can get phosphorylated. That's going to activate or make available the active site for other substrates. So this IRS1 is insulin receptor substrate 1. So that says that over there. And so now that's another protein that is cellular, but it finds its way to the cellular part of the insulin receptor. It gets phosphorylated, and then it goes and does its thing inside the cell and triggers lots of other things to happen. Okay, so this part you don't have to worry about for now. But basically what insulin binding to the receptor does on the outside of the cell has a lot of in-cell effects, but it's mediated through this phosphorylation cascade. Okay, so here is ERK. So all these proteins that end with K, the K stands for kinase. So phosphorylation of one protein turns it on to become a kinase that will phosphorylate another protein, that will phosphorylate another protein. Okay, so kinase, kinase, kinase. Then ultimately, new proteins get made. So that's the one of the downstream effects of insulin. So ignore the details. So what I want you to know today is insulin binding on the outside triggers a phosphorylation cascade inside the cell that ultimately affects downstream detail. Oops. Okay, so here's another one. So here again is our same insulin receptor substrate that gets phosphorylated. So one of the things that happens is phosphatidylinositol gets phosphorylated. So that's a little lipid. And then it triggers opening of a glucose receptor to bring glucose into the cell. So we're going to spend a lot of time talking about glucose for the next couple of weeks. So what does insulin trigger in your body? So if insulin is the ligand, what is the global effect that it's trying to tell your body? So your blood sugar is high. So if you have high sugar content, then you typically have high insulin. So what are you going to do with the sugar that's in your bloodstream? So what does insulin tell your body to do? That's one of the things we're going to see. So that's one of the options that can happen. So if you don't need to use it for energy, you might as well store it for later. Decrease your blood glucose, store the sugar for later. What's something else it could do? Use it for energy, right? So if you eat a Pop-Tart for breakfast, you might need some of that energy, not just to store it for later, but to actually get out of bed and get moving, right? So we're going to open up that glucose channel, bring the glucose from the bloodstream into the cell where it can get metabolized or stored for later. Okay, so here's one thing, synthesis of glycogen, or using the glucose as an energy source. So again, that's a lot of stuff going on. That is just a preview of things to come, but it's triggered by the insulin receptor binding insulin on the outside, causing a phosphorylation cascade inside the cell, and a lot of moving parts. Things happen to either use the glucose as energy or store it for later. Okay, so that's one receptor enzyme that we're going to see again. Another receptor enzyme, so epidermal growth factor. So where's your epidermis? Your skin, right? So it's typical, middle school joke. Hey, your epidermis is showing, right? Your skin is showing. So epidermal growth factor, so that's a hormone. What do you think it triggers? Your skin grows, okay? So as you're growing, you might need more skin, right? And so here is a receptor that binds that epidermal growth factor. It is also has an intracellular tyrosine kinase domain that when EGF binds, it's going to do things inside the cell. It's going to phosphorylate things. What happens, though, if you have a mutant form that is on all the time, even when it's not binding its ligand? So that could be bad. Okay, so it turns out that there are mutations in genes that encode these receptors to have them turned on all the time. So that's called constitutive. I don't know if I spelled that right or not. Constitutive activation. So basically, it's always on. And that's bad because now you have that downstream effect without the signal that's supposed to turn on the downstream effect, and that can result in cancer. Okay, so if you have cells that are growing when they're not supposed to, that's kind of a clue for tumor growth, right? So it turns out that some of the mutations that cause tumor growth are mutations in these signaling enzymes that turn on pathways that are not supposed to be turned on unless there's a ligand there for it. Okay, and in fact, these kinases are so important in cellular regulation and kind of dictating a pathway, but there are some new anti-cancer drugs that are targeting some of these kinase enzymes. The challenge is there are so many kinases in an organism that the challenge is to be very specific, so you only turn off the kinase that's in that pathway, triggering that tumor formation. And the last kind of receptors we'll talk very briefly about are steroid receptors. And so steroids are, well, here's a hormone there, it's not showing you much. Steroids tend to be nonpolar or hydrophobic. So now, instead of being soluble in the cytoplasm and binding to a cytoplasmic protein, steroid hormones can easily go through the membrane because they're nonpolar, but then they have to be bound to a protein to protect them from the aqueous environment. So steroid hormones are most often active in the nucleus, where they can slip easily through the membranes. So here's one receptor that, in fact, the signaling molecule goes into the cell. And it doesn't just go through the cell membrane, it goes through the nuclear membrane, then binds a protein in the nucleus. Many steroid receptors are actually transcriptional factors. They're going to turn on production of a protein. So the steroid hormone gets all the way into the nucleus of the cell, binds its receptor, conformational change, DNA is bound to turn on or turn off gene transcription. So that's how these are different from some of the other receptors. Okay, so it's going to have a slower effect than, say, acetylcholine. So you want adrenaline to be very fast. You want insulin response to be very fast. You want acetylcholine response to be very fast. These nuclear receptors and the steroid hormones, that's going to have a longer lasting effect. And it's going to be slower. It's going to depend on proteins getting made, whereas the others are very rapid response. Okay, so we're going to have some kind of longer-term adaptation in the organism. And that's all we're going to say about signaling. Any questions? So this is just a piece of chapter 12. Okay, and so continue recording. And I should have put all these together, but we're going to jump around. So did anyone have any questions on that? Yes. The chapter that we totally skipped was chapter 11, I think. Oh, that Emil covered? That was chapter 13. So 11 was lipids and membranes. 12 was signaling. I should probably check this, but I think that's right. 13 is introduction to was bioenergetics. 14 is glycolysis, I believe. Yes, Tyler.
The EGFR, it's only a single RTK, right? It's not like the double and single.
It is single. Yep. Yeah. So I don't know that they all are, but I think that's monomeric. Yep, I think so. So did you have any questions on bioenergetics, or should we talk about glycolysis and then we can come back to bioenergetics if you have specific questions later? So when we will certainly use some of those cofactors and enzyme mechanisms, and I'll show you the business end of the molecule that I want you to know. Am I ever going to ask you to draw the whole structure of NAD or FAD? No. Yeah, so the part that actually is involved in redox chemistry is important, but you don't have to know the whole thing. I would say those are things you should recognize, though. Okay, so I guess so we would jump to glycolysis, and then as things come up with bioenergetics, where'd you go? As things come up with bioenergetics, we can answer them, and we can do some practice problems. Okay, what does it say? End of show. Oh dear. You know, you'd think once I said it once, it would remember, but no. Okay, glycolysis, which is chapter 14. So now we get into the really exciting part, I think, of biochemistry. Not that structure isn't exciting, but function and these metabolic enzymes, hopefully you will find exciting. And so if the timing was right, here's, well, maybe not today's lecture, but maybe next week's lecture is when the beer song would have come in, because we are going to talk about brewing, but the timing worked, so that was St. Patrick's Day. Come back. So we're going to talk generally about metabolic pathways, and so kind of what are some big picture ideas before we get to the details of glycolysis. And again, I know many of you have seen glycolysis before, probably in cell bio. So how many of you have learned the names of the enzymes in glycolysis? How many of you have learned the structures along the way? How many of you learned the mechanisms for the enzymes? Oh, in cell bio? Okay, very good. So that's where we will emphasize structures and mechanisms rather than just names, because we're chemists. But before we get there, let's talk about the kinds of pathways we're going to see for the rest of the semester. So, we've certainly talked about individual enzymes. So we can talk about, chymotrypsin, and a standalone enzyme. It has a substrate, it has a product, it has a mechanism. But when we start talking about metabolism, we tend to talk about pathways of enzymes. Okay, and you saw a hint of this when we talked about aspartate transcarbamylase, right? So remember we said that was the committed step for pyrimidine biosynthesis. So that meant it was the first enzyme for which we weren't going to go backwards, and we weren't going to shunt off into anything else. If you do the aspartate transcarbamylase step, you're going all the way to pyrimidines, okay? And so a metabolic pathway is a series of enzyme reactions that all go to a common goal, okay? And it could be that we're going to veer off, and maybe this product can go off and do something else, and many times it won't. It's just on its way to getting where we need to go in that metabolic pathway. So we're going to see that glycolysis is a linear pathway, where if we start with one beginning substrate, so glucose, we're going to go all the way to pyruvate. And we could pull off intermediates along the way at some point, but it's a linear pathway of enzymes. We're also going to see the citric acid cycle. If it has the name cycle in it, you can imagine it's a cyclic pathway. So the TCA cycle, or citric acid cycle. Okay, so that's where the same set of enzymes can act numerous times if we keep feeding in some starting material, okay? But the end product will get us back to the beginning of the pathway. We'll see that. And then there's something that you may have not seen before, which is a spiral pathway. And so in the spiral pathway, that's beta oxidation of fatty acids. So when we get to beta oxidation, you will see that, so this is a fatty acid. So if you have hydrocarbon tail, okay, so that is a fatty acid, right? And those tend to be even numbers of carbon groups. Okay, so we could have a C12, a C16 fatty acid. So the fatty acids that our body makes, consume, and that especially if we eat animal fats, those tend to be even numbered carbon fatty acids. And that's partly because that's how they're made. They're made as 2 carbon groups, and then they're broken down as 2 carbon groups. But if you have a C16 fatty acid, and you're going to break off 2 carbon groups, you have to use the same set of enzymes numerous times. Okay, so in fact, for a C16, you go around 7 times to get 8 2-carbon groups. That'll make sense later. Okay, but that's a spiral pathway, so it's the same set of enzymes, and it's acting on the same substrate numerous times around the cycle. So that's the beta oxidation of fatty acids. So that's just these series of enzymatic reactions. So why would it make sense to use a series of reactions instead of just one, for example? Instead of just one enzyme that would just shatter the whole big molecule. So we can imagine if we have glucose, so if glucose is our starting material for glycolysis, if the ultimate reaction is glucose plus oxygen, so that's oxidation, or we're burning the glucose, we could just burn the sugar. Okay, so you could just light it on fire. So what if that glucose now, instead of being in, or that carbon source, instead of being glucose, or instead of being a sugar that we consume, was a piece of paper, which we also know is glucose monomers. So we could light it on fire. So that's one reaction, and we get to the same endpoint if we fully combust carbon dioxide and water. Does that give us any chemical energy? Usable chemical energy that we can use for work? No. But it'll give us light energy, and it'll give us heat energy, but it's not the kind of energy our bodies want to use. Okay, so that would be a single uncontrolled combustion reaction. It releases energy to get to the same products, but not in a way that's biologically useful. So this series of pathways in a metabolic, so these series of enzymes in a metabolic pathway will give us little small packets of energy. And what kind of energy form is that? So is that light? Is that heat? What should it be in? It's a form of energy that we actually want to use. ATP. Okay, so we're going to see that we're going to produce ATP, or we're going to actually produce more reduced cofactors. So all of these individual oxidation steps, we're not actually using oxygen until the very end of our pathway in our mitochondria. So we're going to use oxygen in the mitochondria, but all the enzymes along the way that are going to oxidize our fuel are going to actually transfer the electrons to cofactors to store that electron potential energy in these reduced cofactors that will then get shuttled off to the mitochondria from where we can get our ATP energy in the presence of oxygen. Okay, so we have controlled energy and multiple steps that can be controlled. Okay, so we may not want to just burn all of our sugar at once. We want to trickle it out when we need it. Okay, so both of those are important features of metabolic pathways. So regulation, we've hinted at when we talked about enzyme regulation, but when we talk about regulation of full metabolic pathways, that allows us to respond to what substrates do we have, what products do we need, do we actually need the output of energy right now, or are we just sitting around watching Netflix and we don't really need a whole lot of ATP energy? So that would be, if we have plenty of our product, maybe we want to inhibit the pathway so we don't waste all those intermediates for something we don't need. Okay, so you've already seen feedback inhibition. So again, in the ATCase example, there are fewer examples of feed-forward activation, but we'll see a couple of them, where the product of one enzymatic reaction in a pathway will actually allosterically activate an enzyme downstream, kind of priming the pump for the substrates that are coming downstream. And you would never take all of product B, you just take a little bit of it, right? So a little bit of product B gets siphoned off, because especially with alloster, you don't need you don't need a lot of it to turn on the enzyme activity for that enzyme. So that's feed-forward activation that we'll see a couple of examples of, but not much. We're going to regulate it. And so what are the catabolic pathways that we're going to see? So really they match the sources of fuel we consume. Okay, so if we consume a diet of proteins, sugars, and fats, then we need pathways to process all of those fuel sources to get us energy. Okay, so proteins will get broken down to amino acids. The bigger polysaccharides will get broken down into smaller sugars. And the triacylglycerols that are in our diet or stored in our cells will get broken down into fatty acids. They're all going to feed into this interconnected set of metabolic pathways in different ways. So what we're going to see for the next couple of classes is just that tiny little piece. I'm sure some of you have seen these big posters of all the metabolic pathways. So it's not even just catabolic pathways, it's DNA polymerase and all kinds of other amino acid metabolism. So that's really impressive. And we're just going to look at a tiny little piece, which is kind of sad because it seems like a big deal now when you have to think about all the molecules you have to learn. But it's so small compared to what your body has to keep track of every minute of every day. So the three nutrients, protein, sugars, fats, all have enzymes, some of which you've seen that get them partly started in metabolism and then more sophisticated enzymes to keep the process going. And you'll notice that there's just a couple of places where ATP shows up. So we get a little bit of ATP from glycolysis. We get a little bit of an ATP equivalent, GTP, from the citric acid cycle, And here's where most of it happens, in oxidative phosphorylation in the mitochondria. So in the electron, so the end product of electron transfer is lots of ATP. Okay, so before we get to specific enzyme reactions, we'll come back to thermodynamics for a minute. So if these metabolic reactions are going to interconvert different forms of the chemistry, we're trying to extract energy from one compound into a form that we can use. Okay, so we're taking the potential chemical energy of glucose and we're going to turn it into ATP energy or reduce cofactors that we can get ATP from later. So these metabolic reactions interconvert matter and energy. And something to think about is that these pathways are maintained at the steady state. So remember we talked about steady state assumption in enzyme kinetics. So it's not exactly the same kind of enzyme, same kind of steady state, but kind of. And so if we have a reaction at equilibrium, that means that the forward and reverse rates are the same. Okay, so once we've reached equilibrium, so maybe not at the beginning, so if we start out with all A, there's some driving force to make some of B, but then we're going to the system's going to be at equilibrium, so the making of B and the making of A are happening at the same rate once we've reached equilibrium. So that's a useful way to think about things, but that's not how metabolism works. So in metabolism, if we need to get to C, we don't really want to go backwards at the same rate we're going forwards, so we call that steady state. So if we have this metabolic intermediate, so molecule B, so now what we want to think about is the rate of change of B should be close to 0. So if we're trying to get to C and we have a bunch of A, we're going to go to B in the meantime, but ultimately we want to get to C. So we're going to keep going through this pathway, and so this is called metabolic flux. So that's the overall rate that you're going through the pathway. And so if you have a lot of A and you're trying to get to C, your flux will increase. Okay, so we're going to increase the rate of that and the rate of that. But for the most part, we're not trying to build up any B. We're just using it to get to C. So there isn't 0 B, but the rate of change of B should be close to 0. So here's my really piddly, sad example. So it's like you have a bucket with a hole in the bottom. And so if the bucket is the pathway, you're putting things in at the same rate you're taking them out. So the hole in the bottom of the bucket should drain the bucket at the same rate that it's being filled. So you never want the bucket to empty, but you don't want it to overflow either. So the metabolic flux is things going in and things going out should be at the same rate. And I know that's very pictorial, fluffy. We'll get to numbers later. Okay, so now we're going to talk about glycolysis. Does anyone have questions about those fluffy concepts in the meantime? I promise we'll get more details and more numbers later. Okay, so when we talk about glycolysis, so that's sugar metabolism. So I think you know that already. But it's also fed by amino acid and lipid metabolism. Okay, so again, we'll talk more about that later. So what does the word ubiquitous mean? So you see it all the time. So most organisms do glycolysis. No. Okay, so in my lab, we actually work with an organism that doesn't have glycolytic enzymes. And that's because it's a parasitic bacteria. So it needs to get its metabolites from the host because it can't make them itself. Okay, so most organisms do glycolysis, but not every. So all differentiated cell types of most multicellular, so definitely multicellular organisms, but some bacteria, they don't do it. It happens in the cytoplasm. So that's something, as we talk about different metabolic pathways, let's remember where things happen. Okay, so up until now, we've been talking about enzymes kind of in isolation. We've assumed that they're cytoplasmic enzymes because we talked about them being in an aqueous environment. We're going to see some enzymes and some metabolic pathways that are not in the cytoplasm. So let's pay attention to where they are. So glycolysis happens in the cytoplasm, and it's pretty efficient. Okay, so if you had a combustion engine that was 60% efficient, you'd be pretty impressed. Okay, so it's not 100%, but it's pretty good. For some cell types and some microorganisms, it's the only source of energy. So for example, we're going to spend a lot of time talking about your brain and how your brain needs sugar, because your brain does glycolysis and it doesn't metabolize fat and it doesn't metabolize protein. So your brain cells need sugar, or they don't get energy, so there's a couple of last-ditch compensation efforts that your brain cells do if sugar is very low, but it's not a good long-term strategy. So your brain needs sugar. Okay, so here we're going to get to what glucose does. So we talked about how we could, if we had glucose, we could store it as glycogen. We could, depending on what kind of organism you are, you could make polysaccharide of your cell wall. You could make the extracellular matrix. So glucose monomers are used for that as well. We're going to see another odd pathway that we can regenerate. We can take those sugars and we can make other things with them. But mostly what we're going to talk about now is oxidizing the glucose to make pyruvate. That pyruvate will feed into then the citric acid cycle. So we're going to get a little bit of ATP. And we're going to get some reduced cofactors, even just in glycolysis. Okay, so glycolysis is only one of the things that can happen to glucose, but it's an important thing. Okay, so the overall reaction of glycolysis is we're going to take a molecule of glucose. It's going to convert two ADP molecules to ATP. So the payoff is two ATP molecules. We're going to get two NADHs, so reduced cofactor. And we're going to get two molecules of pyruvate. So you probably recognize glucose, and this is pyruvate. Okay, so we're actually going to get two of those, right, because glucose is a 6-carbon sugar, pyruvate is a 3-carbon molecule. So we're going to get 2 pyruvates for every molecule of glucose. But there's still a lot of chemical potential in pyruvates, so as it goes through the citric acid cycle, then we're going to extract more energy from that. Okay? And then the standard state free energy change is quite negative. And so from what you guys talked about on Tuesday, what does that mean to you? So what does a negative delta G mean? So it's spontaneous, right? So it's going to go in this direction spontaneously if what? So what is the standard, so this little, what does that mean? What does standard state mean? Yes, Karen. Okay, good. Yes, 25 degrees Celsius. And standard state assumes 1 molar concentrations of substrates and products. So if we start with 1 molar glucose, 1 molar pyruvate, then we're going to go in the direction of pyruvate. Okay, so that's saying the driving force is going to pyruvate if we start out with equal amounts of everything. Yes? So everything is 1 molar under normal standard state. So biological standard state is 1 millimolar magnesium and a pH of 7, which is not 1 molar protons. So that's the concessions for biological standard states. So the prime means standard state, so the prime means biological, the little naught means standard state. Yes. Mm-hmm. Yep. So if magnesium was a substrate or product, then it would not be at 1 molar. If protons were involved, yep, it would not be at 1 molar. Okay, so we're just kind of saying this is a normal system. We're not going to have a pH of -1, right? Okay, so that's the overall reaction, and it's spontaneous in this direction, so that's useful, right? Because we want to actually make products of glycolysis. Okay, come on. But how do we get there? Okay, so glycolysis is 10 enzymes, and it looks daunting now, but I promise it's not that bad. but it's going to take us 10 enzymatic steps to get from glycolysis to 2 molecules of pyruvate. So the nice thing is a lot of these enzymes are repeats of one another. Okay, so kinase, 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 kinase. Okay, so there's 4 kinase enzymes. We're going to see isomerase, mutase, isomerase, right? A dehydrogenase, so what class of enzyme is that? So that's an oxidoreductase. Here's where we're going to see some electrons moving around. And we're running out of symbols. So aldolase is actually a lyase. So we're going to see double bonds formed and made. Okay, so we're going to see each of those individually. And that's just a summary from the book, if it helps you to understand, but we're going to go through them individually. So in glycolysis, we talk about the 2 stages or the 2 phases. And again, if you've talked about glycolysis in another class, you're probably familiar with this. So the first stage is the hexose stage, or it's also sometimes called the investment stage. So hexose because we have 6 carbon molecules. Investment because it's actually going to cost us ATP at the beginning. But then in the triose stage, so these are 6 carbon molecules, these are 3 carbon molecules. The triose stage is also called the payoff because the ATP that we put in the hexose stage is now going to come back to us with interest in the payoff stage. We're going to get, in the end, more ATP than we put in, but we put it in at the beginning and we get more out at the end. Okay? So it's going to cost us ATP at the beginning. It's going to pay off ATP at the end. So we've already seen that we're going to see a bunch of kinase reactions. We're going to see some isomerase reactions. We're going to see an oxidoreductase. We're going to see a couple of lyases. And so there's 3 main mechanisms that we're going to go through that you're going to be responsible for. But I promise they're not that bad. And I'm not making you do the worst one. So the first enzyme of glycolysis is called hexokinase. And we're going to see later on that there's a similar enzyme called glucokinase that basically does the same thing, but in a different cell type, and so we'll kind of keep them straight. So that's in parentheses because we're not going to talk about it now. But hexokinase, you know that kinases add phosphate groups, so it's not too surprising then that hexokinase will add a phosphate to a hexose, and in particular glucose. Okay, so glucose will get phosphorylated at the hydroxyl group on the 6 carbon, and that's coming from a molecule of ATP. So it's coming from so that phosphate group that gets added is the gamma phosphate of ATP, the one that's furthest away from the A base. Anytime there's ATP involved, there's probably some magnesium involved because it has to adjust for those negative charges. So on each of these slides, you're going to see a delta G value. You do not have to learn that number. What I will, eventually we'll talk about which reactions are can go forwards and backwards, and which ones are really only going to go forwards. Okay, so we're going to talk about near equilibrium reactions versus metabolically irreversible. Did you guys talk about this on Tuesday? Okay, we'll get to it later. So when we see standard state free energy changes, so if it's greater than about 6 kilojoules per mole, the absolute value, then it's probably irreversible. So we'll come back to that. Just file that away. Don't think about it too much. So there is hexokinase. So if you want to draw that hexokinase mechanism using the ring form of glucose, that's totally fine. At some point, we're going to be opened up in our sugars, and so we're going to go with the linear form. So I just like to start with the linear form, but you can do it however you want. Okay, so if we have glucose in its linear form, so this is the 6 carbon, so that's the one that's going to get phosphorylated. So the hydroxyl on that 6 carbon will attack that gamma phosphate of ATP. And I think you went through this on Tuesday. So that double bond will open up and then collapse, and then we're going to cleave that gamma phosphate group off to give us now glucose 6-phosphate. Easy peasy, right? Simplest mechanism you'll ever learn. Just going to grab that gamma phosphate off the ATP, and it's going to stay there. Now ADP will go off and do something. Probably get rephosphorylated. Okay, so one mechanism down, very easy. Next one, not quite as easy, but that's okay. So now we have glucose 6-phosphate, and we're going to isomerize it to fructose 6-phosphate. Okay, so here you can see that standard state free energy change is not a big number, either positive or negative. So it's positive, but it's pretty close to 0, right? So it's small, the absolute value is small. And so what this means is it's reversible. That probably is not too surprising. So in isomerase, maybe it's not too surprising that it can go forwards and backwards. Okay, so it's just rearranging internally in the molecule. So the question is, if this is glucose, so it's an aldohexose, we have the aldose group there, and fructose is a ketohexose, so we have a ketone there. So how do we go from an aldehyde to a ketone? So what is the mechanism? So how does that move? Okay, so now we have an isomerase mechanism. So phosphohexose isomerase is that second enzyme in glycolysis. So what happens? So again, I would say you certainly could draw the ring form, but all the chemistry is going to happen to the linear form. So let's just think about the linear form of the sugars. Okay, so we're going to have a base. And in fact, it's glutamic acid, but that's not quite so critical. The textbook draws it just as a general base. So would you predict that hydrogen on the C2 of glucose 6-phosphate is particularly acidic? No, that is not an acidic proton. This is a very bizarre base abstraction of a proton, but that's what the enzyme does. So don't let it bother you too much. It's just what the enzyme does. So that base is going to abstract the proton on that C2 position of glucose 6-phosphate. The electrons then will make a double bond between C1 and C2, which means now we have too many bonds to C1, so we're going to open up that aldehyde carbon-oxygen double bond and grab a proton from solution. So I would say when I would draw this mechanism, I would put a base there and an acid there. So general acid, general base mechanism. Because now that the electrons that had been in that double bond are just going to grab that proton. Okay, so now it's showing the proton just coming in, but let's just put it on a general acid group. So that probably sounds familiar from organic chemistry, a cis-ene diol. So ene, we have a carbon-carbon double bond. Diol, because there's 2 hydroxyl groups. Cis, they're on the same side. Okay, so if that term helps you remember what you're going to, that might be useful. You don't have to remember that it's a cis-ene diol, but sometimes it's useful to say, okay, where am I going? Oh yeah, cis-ene diol. So that's where I have to get to. So now we have this cis-ene diol. We have a protonated general base, and I would say a deprotonated general acid. Textbook doesn't want to show it, but I'll show it anyway. And now we just have to move everything the other direction. Okay, so now our general acid that was deprotonated is going to pick up that a proton, right? So it's not the same proton that it donated. It was a proton that was on the glucose 6-phosphate to begin with. And again, those electrons will collapse. Oh, we're making a new carbon-oxygen double bond. These electrons will grab the proton from the protonated general base to get us back to a deprotonated general base, and I would say a protonated general acid. But now we've converted our aldohexose into a ketohexose. And in fact, it's a phosphorylated aldohexose, so glucose 6-phosphate. Glucose 6-phosphate has now become fructose 6-phosphate. So what mechanistic features are there? Do you see any covalent catalysis? Nope. Really just general acid, general base catalysis. And there also has to be orientation, proximity, transition state stabilization, that's assumed. You don't see any metal ions, so really general acid, general base catalysis is the big one. Make sense? So now you know two mechanisms of glycolysis. The next enzyme, phosphofructokinase 1. So again, it's a kinase, so you can probably predict we're going to take an ATP molecule, grab the gamma phosphate from it, but now instead of going on the hydroxyl in the 6th position, that already has a phosphate group on it, now we're going to use the hydroxyl group on C1 to give us fructose 1,6-bisphosphate. Okay, so the bis means there's two phosphate groups, but they're not connected to each other. So remember when we talked about ADP, adenosine diphosphate? So di also means two, but in a diphosphate, the phosphates are connected to each other. And in a bisphosphate, there's two phosphate groups on the molecule, but they're not connected to each other. Okay, so here, if we were to draw it out in linear form, okay, so 1, 2, so now this is fructose 1,6-bisphosphate. Conveniently, so phosphofructokinase 1, so the 1 means the phosphate group goes on the 1-hydroxyl. Later on in glycolysis, we're going to see a phosphofructokinase 2. It puts the phosphate group on the 2-hydroxyl. Make sense? Again, it's a kinase. We use ATP, we grab the gamma phosphate, that's all you need to know. So you could do that mechanism too. It's the same as hexokinase. So now from fructose 1,6-bisphosphate, so now you can see we have a 6-carbon sugar with 2 phosphate groups, and hopefully you can see then that it might be convenient if we split it into 2 3-carbon groups. Now they both have phosphates on them, right? So that's what aldolase does. So aldolase will take us from, again, I don't know why the textbook likes to draw them in ring form. I find that a little bit less useful. But aldolase is the enzyme that takes us from the hexose stage to the triose stage, because it's going to split the fructose 1,6-bisphosphate. I remember where my hydroxyl groups are. Did I just do that backwards? Nope. Okay, so it's going to split between carbon 3 and carbon 4 to give us 2 3-carbon groups, each of which has a phosphate group on one end of it. Okay, and so the 2 molecules that we're going to end up with are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So you've seen glyceraldehyde 3-phosphate before, and dihydroxyacetone phosphate. So hopefully you remember that this is acetone. And if you don't, now you know. So that's acetone. So dihydroxyacetone would be one of those hydroxyls replaced with a hydroxyl group. And if one of them has a phosphate, it's dihydroxyacetone phosphate. Again, if you remember from organic chemistry some of these names of molecules, that might help you in naming them. If it doesn't, that's fine. You can just start with the molecules you need to know. And then glycerol, so remember that's glycerol. So glyceraldehyde would be one of these being an aldehyde instead of a hydroxyl. And so glyceraldehyde 3-phosphate means start with the backbone of glycerol. One group is an aldehyde, the other end we have the phosphate group. Okay, I'm not going to ask you for the structure of acetone or glycerol. It's just if that helps you to file things away where you already have a slot to put them in, that's great. You are not responsible for the aldolase mechanism. It's a messy one. If you get excited by mechanisms, you're certainly welcome to learn it and understand it. There's just a couple things I want you to remember about aldolase. So first of all, it's going to break carbon-carbon bonds. It is going to use an intermediate that we call a Schiff base intermediate. So if you remember from organic chemistry, a Schiff base is going to be a nitrogen-carbon intermediate. So nitrogen double bonded to carbon. So that's a Schiff base intermediate. That nitrogen is coming from the side chain of a lysine group. So if we have a nitrogen double bonded to carbon and the nitrogen comes from lysine, then you could probably guess it's going to be covalent catalysis, because that nitrogen comes from an enzyme lysine side chain. So aldolase uses covalent catalysis, and it uses a Schiff base intermediate. So the nitrogen from the enzyme lysine is forming that intermediate. And so something else you might remember about it. So it's a ping pong mechanism. Okay, so we're releasing one product. So in this case, we have one substrate and two products, unlike chymotrypsin. But where we do half the reaction, we release one of the products. The other half of the reaction is still covalently attached to the enzyme, so the enzyme hasn't finished its job yet. And then in the second step, it gets cleaved off. Okay, so we end up with glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So where do those carbons go? So when aldolase cleaves between C3 and C4, 1, 2, and 3 carbons go to dihydroxyacetone phosphate, 4, 5, and 6 go to glyceraldehyde 3-phosphate. So some mean graduate school instructor someday may say, if you consume glucose with a C13 label at the C2, where does it end up after the citric acid cycle? I promise I won't ask you that, but here's how you could start to trace it. Okay? So what we're going to see next, I will do one more reaction, is we see our next isomerase, because in the end we get to 2 molecules of pyruvate, so we want to kind of consolidate dihydroxyacetone phosphate and glyceraldehyde 3-phosphate to make them the same molecule. Okay, so what we're going to end up with is we're going to lose our dihydroxyacetone phosphate because it's going to become glyceraldehyde 3-phosphate. Okay, so again, it's just an isomerization reaction. Now we're going from a ketone to an aldehyde. So same idea as before. We're going to have a general base. We're going to have a general acid. We're going to make a cis-ene-diol intermediate. To interconvert the ketone to the aldehyde. And again, delta G is a small number. It's easily reversible. So hopefully what you could see, so that one looks like it would never happen. So a very positive standard state free energy change. We're going to see it's okay, so don't panic, it really does happen. It happens because, so think Le Chatelier's principle, we're pulling off one of the products, and so that'll drive the reaction forward. So all the kinases are going to have pretty negative delta Gs because we're using the ATP energy. That one looks like it wouldn't go forward, except we're going to drive it forward by getting rid of one of the products. And the isomerase is pretty close to 0. It's easily interconvertible one way or the other. Okay, so that's triose phosphate isomerase. And at that point, we have 2 molecules of glyceraldehyde 3-phosphate that will carry us through the rest of glycolysis into pyruvate. Okay, and I'm going to stop a few minutes early. So that was the triose stage. That was the hexose stage. And now the next is we're going to actually get the payoff. So we're going to get NADH. I don't know why I didn't put the ATP in there. We're going to get the ATP. And so that's the payoff. But the next mechanism is one that I want you to learn, and it's not simple, and your brains are not going to pay attention anymore. So we will stop there, and we'll pick up on Tuesday with the triose stage of glycolysis. So if I didn't give you back a quiz and a double helix and you want to get it, come get it now.
I forgot about the disease paper.
Okay. Email it to me in the next couple of days. Hopefully by the end of the weekend. Okay.
Is this the question she said?
Yes. So what I said was anybody who's here got at least a point, and you will see the material again. So you emailed your double helix, and I haven't finished grading them yet, so I will give it to you.
Okay, great. Thank you.
Thank you. You're here on Tuesday, and you emailed your paper, then I will email it back to you.
Thank you.
Have a great time.
Question?
Yeah.
Oh, quiz. I'm sorry. More here, so it's better than a long shot.
Thank you.
I think what I didn't realize there was since I saw we were like writing ourselves.
How do you type again?
Okay.
Yep.
Do you want me to-
You were the only one who did it that way, and I'm like, I don't think you should.
Yeah.
Please do say if you can.
Okay, thank you.
Yep.
I emailed the double helix.
Yes, I'll email it back to you. I haven't graded those yet. I was trying to do this on a plane, and I didn't get to this. Oh, okay. I forgot to let you
But I
know.
just have one more question. Looking at the syllabus, it said that the quizzes are out of 20 points, and we've taken 3, and the total
And it
is
adds up to
18.
18.
So I was just confused.
We will have one more quiz, and it will be out of 18. So that should help the ones who didn't do so well in this one.
Wait,
So-
it'll be out of 18?
Well, so we'll have another quiz. So the total number of quiz points will be more than 18,
Okay.
but
Okay.
there'll be some. We're gonna throw away some of those quiz points. Yep, it'll work out okay, I promise. Yep. I thought I hinted. Apparently, I didn't hint very well.
Literally, where I think recorded was like before the test, which was like I got some time. But I
Yeah.
should have known.
But then the email was like, "We finished." And it's like, yeah. Yeah. Well.
I did have a question.
Uh-huh.
The 3 glyceraldehyde phosphate. Is that what it's called?
Glyceraldehyde 3-phosphate.
Yes.
Uh-huh.
Was that when you were giving examples of the different types of enzymes? There was a pyruvate, and it did an immune transfer. And giving examples of transfer.
So pyruvate is the alpha keto precursor of aspartic acid, maybe? I forget.
Okay, so it was pyruvate,
So the transferase,
the transfers.
I think that's what it was.
Because I knew I recognized one of those. I mean, pyruvate because it's pyruvate, but I
Right.
thought the 3-glyceraldehyde might have been.
Oh, I can't remember.
I can look back.
Where is my recording thing?
The transfers example was from glycolysis or not, but
No, that would have been from amino acid metabolism.
okay.
And
Does it have glycine?
yeah, so it's the precursor of glycine.
Okay.
Sorry. Yeah, so pyruvate is the alpha keto that corresponds to glycine. So if you take an amino group and put it on in place of one of the ketos, it's glycine.
Oh, okay, that makes sense. And my other question, do we need, for the 2 mechanisms that we did today, we didn't talk about their active sites or their...
Nope. Yeah, so it's just, so certainly for the kinases, they're not covalent catalysis, and so there's no enzyme active site side chain that's acting as a nucleophile. It's just positioning.
Gotcha.
Okay.
Okay.
Yep. Where's my recording? Thanks, you too.